Hello everyone. In today's Biology MCQ session, we are discussing important questions from the chapter Biotechnology, Principles and Processes. Question number 1. Identify the correct statement regarding restriction enzymes. They cut DNA randomly, they recognize specific palindromic sequences, they synthesize DNA fragments, they replicate plasmids. The right answer is option B. Restriction enzymes recognize specific palindromic nucleotide sequences in DNA. They act as molecular scissors, cutting DNA at defined sites. Question number 2. The enzyme used to join DNA fragments during recombinant DNA technology is DNA polymerase, RNA polymerase, DNA ligase, restriction endonuclease. The right answer is option C. DNA ligase joins DNA fragments by forming phosphodiester bonds between adjacent nucleotides, sealing nicks in DNA. Question number 3. In recombinant DNA technology, the vector must have origin of replication, selectable marker, multiple cloning site, all of the above. The right answer is option D, all of the above. A vector must contain origin of replication, selectable markers and cloning sites to allow replication and selection of recombinant cells. Question number 4. Assertion. Plasmids are widely used as vectors in genetic engineering. Reason. Plasmids replicate independently of chromosomal DNA. Both assertion and reason are true and reason explains assertion. Both assertion and reason are true but reason does not explain assertion. Assertion true, reason false. Assertion false, reason true. The right answer is option A. Both assertion and reason are true and reason explains assertion. Plasmids possess independent replication ability, allowing inserted genes to multiply inside host cells, making them ideal vectors. Question number 5. The enzyme that synthesizes DNA using RNA as a template is DNA polymerase, reverse transcriptase, RNA polymerase, primase. The right answer is option B. Reverse transcriptase synthesizes complementary DNA from RNA template. It is commonly used in cDNA library preparation. Question number 6. Restriction enzymes were first isolated from dash, viruses, protozoa, fungi, bacteria. The right answer is option D, bacteria. Restriction enzymes were first discovered in bacteria where they act as defense mechanism against bacteriophages. Question number 7. EcoR1 restriction enzyme recognizes the sequence GAATTC, GATATC, GGATCC, AAGCTT. The right answer is option A. EcoR1 recognizes the palindromic sequence GAATTC and cuts between guanine and adenine, producing sticky ends. Question number 8. Which technique separates DNA fragments based on size? PCR, gel electrophoresis, centrifugation, chromatography. The right answer is option B. Gel electrophoresis separates DNA fragments according to their length and charge as they migrate through agarose gel. Question number 9. The thermostable DNA polymerase used in PCR is obtained from Escherichia coli, Bacillus subtilis, Thermus aquaticus, Streptococcus. The right answer is option C, Thermus aquaticus. Taq polymerase from Thermus aquaticus remains active at high temperatures used in PCR denaturation steps. Question number 10. The amplification of DNA in vitro is carried out by DNA ligase, hybridization, restriction digestion, PCR. The right answer is option D, PCR. Polymerase chain reaction amplifies specific DNA sequences using primers and thermostable DNA polymerase. Question number 11. The first genetically engineered human insulin was produced by dash, Eli Lilly Company, Pfizer, Bayer, Roche. The right answer is option A. Eli Lilly Company produced recombinant insulin called Humulin using genetically modified E. coli. Question number 12. Which step in PCR involves separation of DNA strands? Annealing, extension, denaturation, replication. The right answer is option C. During denaturation, the DNA is heated to about 94 degrees Celsius, causing hydrogen bonds to break, separating double strands. Question number 13. The term bioreactor refers to device for DNA sequencing, vessel used for large-scale culture of microbes, enzyme purification system, genetic mapping instrument. The right answer is option B. Bioreactor is a large vessel where microorganisms or cells grow under controlled conditions for product formation. Question number 14. Stirred tank bioreactors are preferred because they maintain oxygen supply, maintain temperature control, allow proper mixing of nutrients, all of the above. The right answer is option D, all of the above. Stirred tank bioreactors provide optimal aeration, mixing, temperature control and pH regulation for optimal microbial growth. Question number 15. Match the following. Column 1, EcoR1, Taq polymerase, DNA ligase, PCR. Column 2, DNA amplification, recognition of palindromic sequence, formation of phosphodiester bond, thermostable enzyme. The right answer is option A. EcoR1 recognizes palindromic sequence, Taq polymerase is a thermostable enzyme, DNA ligase forms phosphodiester bonds, and PCR leads to DNA amplification. Question number 16. Identify the incorrect statement regarding cloning vectors. They should replicate within host cells. They must contain selectable markers. They must destroy inserted genes. They should be easily isolated. The right answer is option C. They must destroy inserted genes. Vectors must preserve inserted genes, not destroy them. They help in replication and expression of foreign DNA. Question number 17. The antibiotic resistance gene in vectors is mainly used for killing host cells, identifying transformed cells, producing antibiotics, DNA replication. The right answer is option B, identify transformed cells. Selectable markers like antibiotic resistance genes helps to identify cells that have successfully taken up recombinant DNA. Question number 18. Which enzyme synthesizes RNA primers during DNA replication? Primase, ligase, helicase, topoisomerase. The right answer is option A. Primase synthesizes short RNA primers that provide 3'OH group for DNA polymerase to start replication. Question number 19. Assertion. Blundins facilitate recombinant DNA formation more efficiently than sticky ends. Reason. Sticky ends can pair with complementary DNA sequences. Both assertion and reason are true, and reason explains assertion. Both assertion and reason are true, but reason does not explain assertion. Assertion true, reason false. Assertion false, reason true. The right answer is option D. Sticky ends form complementary base pairing, making ligation easier and more efficient than blunt ends. Therefore, assertion is false and reason is true. Question number 20. The selectable marker in PBr322 plasmid includes ampicillin resistance gene, tetracycline resistance gene, both A and B, none of these. The right answer is option C, both A and B. The plasmid PBr322 contains ampicillin and tetracycline resistance genes used for selection of recombinants. Question number 21. The enzyme that unwinds DNA during replication is dash, ligase, helicase, polymerase, transcriptase. The right answer is option B. Helicase breaks hydrogen bonds between base pairs, unwinding the DNA double helix. Question number 22. Which vector is commonly used for cloning large DNA fragments? Plasmid, bacteriophage lambda, cosmids, all of the above. The right answer is option D, all of the above. Plasmids, bacteriophage vectors and cosmids are used depending on the size of DNA fragments to be cloned. Question number 23. The process of introducing recombinant DNA into host cells is called transformation, transduction, translation, replication. The right answer is option A. Transformation refers to uptake of foreign DNA by host cells such as E. coli during cloning experiments. Question number 24. In biotechnology, downstream processing refers to DNA replication, isolation and purification of products, gene cloning, DNA sequencing. The right answer is option B, isolation and purification of products. Downstream processing includes recovery, purification, and quality control of the final biotechnological product. Question number 25. Which organism is most commonly used as host in cloning experiments? Streptococcus, Bacillus, Escherichia coli, Mycoplasma. The right answer is option C. Escherichia coli is widely used because of its rapid growth, simple genetics, and widely studied genome. Question number 26. The main advantage of PCR is rapid DNA amplification, DNA sequencing, DNA digestion, protein synthesis. The right answer is option A. PCR enables rapid amplification of specific DNA sequences within a few hours. Question number 27. Which step follows PCR amplification? DNA hybridization, DNA translation, gel electrophoresis, DNA transcription. The right answer is option C, gel electrophoresis. PCR products are usually analyzed using agarose gel electrophoresis to confirm amplification. Question number 28. Identify the correct order in recombinant DNA technology. Isolation, ligation, transformation, expression, transformation, ligation, isolation, ligation, isolation, transformation, digestion, isolation, restriction, digestion, ligation, transformation. The right answer is option D. Isolation, restriction, digestion, ligation, transformation. The DNA is first isolated, cut with restriction enzymes, ligated into vectors, and then introduced into host cells. Question number 29. Assertion. Bioreactors increase product yield. Reason. They maintain optimal environmental conditions for microbial growth. Both true and reason explains assertion. Both true but reason not explanation. Assertion true, reason false. Assertion false, reason true. The right answer is option A, both true and reason explains assertion. Bioreactors control temperature, pH, oxygen and nutrients, enabling maximum microbial productivity. Question number 30. Match the following. Column 1. Restriction enzyme, ORI, PBr322, downstream processing. Column 2. Product purification, initiation of replication, specific DNA cleavage, multiple antibiotic resistance genes. The right answer is option B. Restriction enzyme performs specific DNA cleavage. ORI initiates replication. PBr322 contains multiple antibiotic resistance genes used as selectable markers, and downstream processing involves product purification. Question number 31. The ORI site in a cloning vector refers to site where restriction enzyme cuts, promoter region, antibiotic resistance gene, origin of replication. The right answer is option D. Origin of replication allows the plasmid to replicate inside the host cell, determining copy number. Question number 32. The single-stranded DNA overhangs produced after restriction enzyme digestion are called dash. Blunt ends, sticky ends, complementary ends, overlapping ends. The right answer is option B. Sticky ends are short single-stranded extensions that can pair with complementary sequences, facilitating ligation. Question number 33. Which of the following vectors can carry the largest DNA insert? Plasmid, bacteriophage, yeast artificial chromosome, cosmid. The right answer is option C. Yeast artificial chromosome can carry very large DNA fragments up to 1 megabase, making them useful in genome mapping. Question number 34. Assertion. Agarose gel is commonly used in electrophoresis for DNA separation. Reason. Agarose forms a porous matrix allowing DNA fragments to migrate. Both assertion and reason are true and reason explains assertion. Both assertion and reason are true but reason does not explain assertion. Assertion true, reason false. Assertion false, reason true. The right answer is option A. Both assertion and reason are true and reason explains assertion. Agarose gel creates pores through which DNA fragments move under an electric field, allowing size-based separation. Question number 35. In PCR, the step where primers bind to template DNA is called extension, replication, denaturation, annealing. The right answer is option D, annealing. During annealing, that is at 50 to 65 degrees Celsius, primers bind to complementary sequences on the DNA template. Question number 36. Identify the incorrect statement regarding recombinant DNA technology. It involves combining DNA from different organisms. It requires restriction enzymes and ligase. It occurs naturally in all organisms. It produces genetically modified organisms. The right answer is option C. It occurs naturally in all organisms. Recombinant DNA technology is artificially performed in laboratories and does not occur naturally in all organisms. Question number 37. During gel electrophoresis, DNA fragments move towards cathode, anode, both directions, neutral point. The right answer is option B, anode. DNA is negatively charged due to phosphate groups, so it migrates towards the positive electrode, anode. Question number 38. Identify the correct statement regarding palindromic sequences. They read differently in opposite directions. They occur only in RNA. They read the same in 5' to 3' direction on both strands. They prevent restriction enzyme activity. The right answer is option C. Palindromic sequences read identically in 5' to 3' direction in both the strands, allowing restriction enzymes to recognize them. Question number 39. Which of the following enzymes is responsible for cutting DNA at specific recognition sites in recombinant DNA technology? DNA ligase, restriction endonuclease, reverse transcriptase, DNA polymerase. The right answer is option B. Restriction endonucleases recognize specific palindromic sequences in DNA and cut the strands at those sites, producing sticky or blunt ends. Question number 40. Which of the following is the correct function of DNA ligase in recombinant DNA technology? Cutting DNA at specific sites, unwinding the DNA helix, synthesizing RNA primers, joining DNA fragments by forming phosphodiester bonds. The right answer is option D. DNA ligase joins DNA fragments by forming phosphodiester bonds between adjacent nucleotides, sealing nicks in sugar phosphate backbone during cloning. Thank you for watching the video. Subscribe to the channel to learn more biology MCQs.